drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there. All right, Professor Rebecca Haeusler, thank you for joining me.
Thank you so much, Nick. Glad to be here.
Do you want to start off by telling everyone a little bit about who you are and what your lab studies?
Sure. I am an associate professor at Columbia University in the departments of medicine and pathology and cell biology. I'm also affiliated with our Naomi Berry Diabetes Center and our Digestive and Liver Disease Center. My lab studies insulin signaling and metabolism and how those processes become impaired in metabolic diseases like diabetes or metabolic liver diseases.
Yeah, and one of the things that something about is something that I know very little about, and I don't think a lot of people know about this topic, but it actually ends up being something pretty common for people to have to deal with at some point in their lives, and that's bile acid. So let's start really basic with this stuff because I haven't covered it on the podcast before, and I don't really know much about this myself. But what is bile acid and what does it do?
Bile acids are the primary degradation product of cholesterol. So they are a molecule that allows us to excrete cholesterol. And about half of the cholesterol that is excreted from our bodies is excreted in the form of bile acids. But they also have their own very important roles in digestion and in cellular signaling processes.
And so why would the body need to excrete cholesterol? Why does that need to happen?
Yeah, so unlike glucose or fatty acid or other carbon sources. We cannot burn cholesterol to make energy or heat. If we want to eliminate cholesterol from the body, it has to be excreted, and it is primarily excreted via the hepatobiliary route, such that excess cholesterol is taken up into hepatocytes and then effluxed out the apical side of the hepatocytes into the bile. And then after a meal, the bile travels down the intestine, and some of that cholesterol will be reabsorbed. Actually, much of that cholesterol will be reabsorbed, and some of it will be excreted through the feces.
Got it. So we can't burn off cholesterol for energy like we can glucose or fatty acids. And bile is one way of regulating how much cholesterol is in the body. Basically, if there's too much, we can get rid of some of it, and we can recycle it if we want to recover some of that. So it's basically a way to regulate how much cholesterol is inside of us.
That's right.
And so where is bile made in the body, and like where is it secreted, and where is it entering the GI tract? Give us a basic sense of the spatial landscape there.
Yeah. So the bile forms in the liver from constituents that are secreted from the hepatocytes and the cholangiocytes, two cell types that face the bile. And so bile acids are formed in the hepatocytes and then secreted out the canalicular membrane of the hepatocytes. Hepatocytes, like other cell types, are polarized. So they have one face that faces the blood and one face that faces the bile. And so bile acids are secreted out the canalicular membrane of the hepatocytes into the bile canaliculi. And then the additional biliary components, including some phospholipids, some other inorganic materials, some other toxins, are also excreted out that membrane. And then water and electrolytes are secreted from hepatocytes and from the cholangiocytes that line the bile ducts. Then the bile travels down the bile ducts, and a portion of the bile is stored inside of the gallbladder. Only about half, this is a little bit of a misconception, but only about half of the bile is stored in the gallbladder after an overnight fast. And the rest of it continues down the bile duct and enters into the duodenum. And that is the upper portion of the small intestine.
Got it.
And after a meal, the gallbladder contracts, and that spills an additional bolus of bile down into your duodenum to aid in digestion.
Got it. So like hepatocytes, these are liver cells. So bile acids are made in the liver, and then there's a duct that sends them either to the gallbladder or right to the small intestine. So the bile is made in the liver. Some of it's going to the small intestine. Some of it goes to the gallbladder and just sort of sits there, I would imagine, for some time until it's needed by eating food, basically. And then that will also go into the GI tract.
That's right.
Got it. Okay. And so the bile duct, is it literally like a tube connecting the liver to the gallbladder and to the intestine?
Yes, it's literally a tube, and upstream of the gallbladder there would be like ductules, smaller duct like formations that would collect the bile and the bile acids from all throughout the liver.
Yeah. Okay, so bile is one way we can get rid of excess cholesterol / I don't know if I completely want to say that, right? It's a way of regulating how much cholesterol is in the body, because we excrete some, but then we recover some of it. Like that's kind of also like how the kidneys and other things work. Like you're excreting and recovering it. Like just it's a way to cycle and fine tune how much cholesterol, I guess, stays in the body versus gets excreted. We have to do that, you said, because we can't burn cholesterol for energy like we can other things. And then there's got to be more to it than that, because I know that people often talk about the gallbladder being important for secreting bile after meals. And like the one thing I know here is that apparently this bile is used as an emulsifier. So is that true? And what exactly is that?
Yeah, so the bile is important to emulsify the food that we have consumed, especially the fatty food that we have consumed. So lipids, when we eat them, would be present in like an insoluble fat globule in the upper intestine if we couldn't emulsify it into smaller lipid droplets, what we call them mixed micelles, which aid in their absorption. So by emulsifying the lipid that we've eaten, we increase the surface area of the lipid in the meal such that the pancreatic lipases can more efficiently hydrolyze all of the triglycerides that need to be hydrolyzed in order to absorb that fat. We cannot absorb triglycerides intact. We have to hydrolyze them into smaller constituents like monoacylglycerols and free fatty acids. So the bile acids and the phospholipids that are contained in the bile are required to form those mixed micelles that can encapsulate the dietary lipid.
Got it. So we want to be able to digest our food, obviously. We're eating a bunch of different stuff. Some of it's dissolvable in water. Some of it's fatty. You can't mix oil and water together easily. So I guess if someone imagines pouring something like olive oil into a cup of water, it would naturally just be like a blob of oil sitting at the top, and it wouldn't mix together in the water. And so the surface area of that oil that would be touching the water is relatively small. And so if you were to say shake it up violently, you could maybe disperse that one big blob of oil into many, many, many tiny blobs of oil. So now there's no more points of contact between the water and the oil. And in the case of our digestion, the reason you sort of want to do that, you want to emulsify things or mix the oils and the waters together, is basically to increase the surface area of all the stuff that will get chopped up by digestive enzymes.
Yes, that's essentially it. We also have lining the inside of the GI tract something called the unstirred water layer. And the polar bile acids and phospholipids help ferry those neutral fatty acids across that unstirred water layer to be absorbed into the enterocytes.
Yeah. Can you talk a little bit more about the bile acid synthesis in the liver? So there's cholesterol contained in the bile acid, and I guess we can think of that largely as excess cholesterol that doesn't need to be sent somewhere else for other purposes. But what is the bile acid actually made out of? Like what are its core components? What's feeding into the pathway that leads to bile acid?
Yeah, the core component really is cholesterol. And then there are about a dozen different enzymes that act to modify that cholesterol to become a bile acid.
Got it. And so what is, when we start thinking about the interaction between bile acid production and secretion and eating meals and the emulsification side of this, what are sort of the stimuli in the form of foods like sugars or fatty acids and in the form of hormones like insulin that regulate bile acid production and secretion? What are sort of the main things there that are regulating this from an input standpoint?
One of the strongest regulators of bile acid production are bile acids themselves. So bile acids are cytotoxic. They can be toxic to cells when they are inside the cell because they can emulsify lipids, for example, membrane lipids.
Got it. So you could potentially destroy the lipid structures in your mitochondria or other things inside of cells if too much bile acid actually gets in there.
Yes. So bile acids have a strong ability to suppress their own synthesis in the hepatocytes. And they also have a strong ability to promote their own efflux out of hepatocytes and into bile. But bile acid production can also be regulated by other hormones and other signaling pathways. And the bile release is also regulated by hormones and signaling pathways. And one of the best known hormones for promoting gallbladder contraction is cholecystokinin, CCK, which is secreted from the intestine in response to a meal, and it causes the gallbladder to contract and release the bile down into the intestine.
Got it. Okay, so bile acids are good emulsifiers, but that has a good side and a bad side. You can emulsify the fats in a bolus of food that you're eating, which is good because it's going to aid digestion, especially if that that bolus of food contains a lot of fatty substances mixed with non-fatty substances, but you can have too much bile acid because we've got a lot of lipids we don't want to emulsify in our body, our cell membranes or mitochondrial membranes and so forth. So bile acid regulates itself. When it goes up, it can sort of downregulate its own production so that we don't get too much, so that we don't start emulsifying all of our own cellular lipids that we do not want to emulsify.
That's right. And some very cool imaging studies have been done using a probe that is a labeled bile acid, showing that essentially the maximum time for an individual bile acid inside of a hepatocyte is only 2.5 minutes. So the regulation of the production and the transport of bile acids across cells is really essential to protect the cell.
Got it. So the cells don't want to touch or handle the bile acids for too long. I would imagine that's a general thing. You don't want bile acids to get into most of your cells, and that also probably the gallbladder has specializations to sort of hold and contain it.
That's right. The gallbladder and the bile itself, the cholangiocytes, are protected from the bile acids due to the so-called bicarbonate umbrella. And the other cell types that experience a lot of bile acids are the hepatocytes and the enterocytes, the absorptive cells of the intestine, because each bile acid in our body is going to pass through those cells multiple times per day. And so those cells, I consider them like professional at handling bile acids, and they have tight control over the transporters and the binding proteins that handle bile acids.
Yeah. And so we don't want to have too much bile acid because too much of an emulsifier puts in jeopardy our own cell membranes and the lipids inside of our cells. What about too little bile acid? Does that happen very often? What happens if we're not sort of secreting or producing enough of it?
That does sometimes happen especially in like genetic conditions. There are individuals who would have rare mutations in, for example, the rate limiting step of bile acid synthesis. And in that case, if you simply had too little production of bile acids, you can have some complications, including some things like excess high cholesterol, high LDL cholesterol. And you can also have cholesterol gallstones, where the cholesterol in the bile is not sufficiently solubilized because there are insufficient bile acids, and then that can cause gallstones. And then you can have malabsorption in the intestine with insufficient bile acids.
Got it. So that stuff can happen, but I'm guessing it's more common to have problems on the too much bile acid side.
Yep.
And is this sort of why like people get their gallbladder removed? That's not that uncommon. And it's, we'll talk about that more later maybe, but it's not catastrophic to have your gallbladder removed. Like it can happen and you'll live. Is that basically why? Because too much is more problematic than too little when it comes to bile?
This is also a common misconception. People get their gallbladder removed not because they have excess bile acids, but usually because they have gallstones. And so inside the gallbladder, the bile gets very concentrated, and including the cholesterol in there. And if the proportions of bile acids, phospholipids, and free cholesterol are off, and there is too much cholesterol relative to the emulsifying components, the bile acids and the phospholipids, then the cholesterol can form stones that in that concentrated setting of the gallbladder or the bile duct can then be very painful.
Got it.
So people get the gallbladder removed to eliminate the concentrating effect and eliminate the gallstones. But they would still continue to produce bile, continue to produce bile acids, which would then just continue traveling down the bile duct. And so they would ultimately have less like pulsatility in terms of bile release into the intestine, although there's still some pulsatility because there's still a sphincter that controls.
Yeah. Okay. So this makes sense. I was wondering about this. So if you have your gallbladder removed, I think the piece I was missing before talking to you was you have these ducts that take the bile from the liver to the gallbladder and also in parallel the intestine. So if you remove the gallbladder, you're still making all the bile acid in the liver, and it can still get into the GI tract via these ducts, but it can't be sort of held and secreted by the gallbladder. The reason people tend to get the gallbladder removed is gallstones. So basically, if there's too much cholesterol in there, it's almost like I don't know. It's like a solution that's been super saturated and some of it just falls out of solution. There's too much of some component in there, and it creates these gallstones, which are probably painful, I'm guessing, and problematic in other ways. So you can get your gallbladder removed and be relatively okay. You're still making the bile acid. It's still going from the liver to the small intestine.
Yes, exactly. You've got it.
So what tends to be the reasons people need to get that gallbladder removed? In other words, why is the proportion of cholesterol off in the first place that's then giving them the gallstones?
That's a good question that I don't have a great answer for.
Yeah. Is that the type of thing that we probably don't have a great answer for in general?
It's possible. I mean, there are certainly some conditions that are that are described, let's say. For example, people sometimes, if they lose weight very fast, but then like connecting the molecular or like chemical mechanisms as to why the bile became supersaturated with cholesterol.
Yeah.
Don't think that is actually understood. But yeah, there may be other conditions that are better understood that I am not familiar with.
Yeah. Okay, so we've got this parallel pathway from the liver to the gallbladder and these ducts to the small intestine that bile can go through. Could we think of the purpose of the gallbladder as being like, it's just sort of holding on to additional bile acids so that if you need a bigger injection because you had a particularly fatty meal, that's when that would be secreted from the gallbladder?
That's right. Yep.
Got it. So there's always going to be some coming through because there's always going to be some baseline level of emulsification you probably need no matter what you're eating. There's always going to be some amount of fat in there, but sometimes obviously we eat more fat than we do at other times. And so we might need this extra injection. That's basically the gallbladder.
That's right.
Okay. And when we start to think about comparing healthy bile production, healthy gallbladder function, all of this stuff to states of metabolic disease, what do we start thinking about here in terms of how some of the common metabolic diseases affect things like bile acid secretion and production and all the stuff we've talked about already? I know, for example, that you've done a lot of work to do with insulin resistance and type 2 diabetes. How does that condition start to tie into the bile acid production and some of the biology we just talked about?
Yeah. So people with a variety of different kinds of metabolic and liver diseases can have changes in the bile acid production and transport. And in my lab, we have particularly focused on insulin signaling and insulin resistance and how those processes control bile acid production, composition, and transport. And so, for example, one of the strongest connections that we have made is the connection between insulin signaling and a bile acid modifying enzyme called CYP8B1, which is a sterol 12 alpha hydroxylase. It adds a hydroxyl group to the carbon 12 of a bile acid and ultimately produces a certain family of bile acids that all have a hydroxylation group in the carbon 12 position. And by modifying the types of bile acids that are being produced in the liver, it changes the total, it changes the effect, the consequences, the effects of bile acids, because different bile acids can carry out their emulsification and their signaling functions differentially due to their different chemical structures.
Mm-hmm, mm-hmm. And so what, like in general, when we think about type 2 diabetes, how are these things working differently in general in that population versus a non-diabetic population?
There have been a number of investigators studying the links between diabetes and insulin resistance and bile acids. And in general, it is thought that bile acid most studies have observed that bile acid synthesis and particularly the synthesis of the 12 hydroxylated bile acids is increased in the setting of insulin resistance and type 2 diabetes.
Mm-hmm. And so I really don't know this field at all. So help me start to think about this. So if you're type 2 diabetic, you're usually going to be insulin resistant. So your cells aren't responding to insulin. And so they're not sucking in glucose into cells from the bloodstream. So you tend to have elevated glucose. Why would you think there would be higher bile acids in these people? Do they have some need to like emulsify food going through the intestine more?
This is a great question, and it's ultimately a philosophical one.
Yeah.
It's something that I and other people working in this area have thought about a lot. And one of the possibilities is that it goes back to the possibility that insulin resistance arose as a response that is beneficial in some settings.
Right.
Like
you can think of it as you can think of it as an adaptation.
As an adaptation.
Yeah.
And so if insulin resistance is an adaptation that is particularly useful during starvation, then you might think if you were a starving individual that you would want to then absorb everything that you were taking in.
Right. Therefore, you'd want maximum emulsification so that you could, chop up every piece of fat and other things that are in the food as possible.
Yes.
Got it. Okay, that's one way to potentially think about it.
Yep, that's one way.
Got it. What is when people have their gallbladders removed? So we already said that doesn't kill bile acid synthesis. You're still making it in the liver. It's still going through the duct to the small intestine. But if you don't have your gallbladder, are there any like major problems that come from that? Do you just have sort of a lower tolerance for fatty food?
Yeah, a lower tolerance for fatty food. Yep.
Yeah.
Mm-hmm.
Okay. So yeah, you basically just can't emulsify it as well. It's not that you can't emulsify it at all. You can't emulsify it as well. So if you were to eat particularly fatty food, I would imagine that you're just not digesting it as well because you're not emulsifying it. So the enzymes aren't chopping it up as much, and therefore you're not absorbing all that fatty stuff as readily.
Indeed.
Got it. And so emulsifiers just allow you to mix fats and non-fats together. We have this natural emulsifier in the form of bile acid, and that's basically what we've talked about so far. There's a lot of emulsifiers used in foods. What's your general perspective on the emulsifiers in processed foods today? If you can have too much bile acid and too much emulsification, and we said that the bile acids can be cytotoxic, can you have too much of these other emulsifiers, and can those be problematic in terms of what they're doing in our GI tract?
I don't know the answer to that, but it would depend on whether or not they can get into cells.
Got it.
One of the features of bile acids is that we have active transporters that are pulling them into our enterocytes and our hepatocytes. So there is a major transport of bile acids into those cells, and therefore those cells have to deal with them. So in terms of the emulsifiers that are put into processed foods, I don't know enough about them to know whether or not they are, how well they're getting incorporated into our cells.
Yeah, so there can be a cytotoxic effect of bile acid, our natural emulsifier, if it gets inside of cells. Are there any problems that can come from having a lot of bile acid secretion just because of when you think about the emulsifying property of any emulsifier, including bile acid, can you have too much and it messes with like the fatty mucus lining of your GI tract and things like that?
The fatty mucus lining, I'm not sure I am aware of information about the effects of bile acids on the mucus itself, but that seems possible.
Yeah.
Yeah.
Got it. Let's talk a little bit more about the regulation of bile acid production by hormones like insulin. When we have high insulin levels and or insulin resistance, What else can you tell us about how that affects things like bile acid synthesis and like what it's doing? Do we know, for example, if people have been like diabetic for a long time, do they start to have problems with bile acid synthesis and secretion? Does it, like you said earlier, that it can go higher, but like can long-term insulin resistance sort of break synthesis or secretion in a more permanent way?
That's an interesting question. I'm not sure there have been longitudinal studies the way you're suggesting, or even cross-sectional studies across different times since diagnosis. But that is an interesting possibility. What we have observed is an increase in the rate of synthesis and this 12 hydroxylation and impaired transport in people that have insulin resistance.
Yeah, yeah. What about things like activity levels, exercise, just physical activity levels? Does that regulate bile acids at all?
Yes, there are a number of investigators working on connections between exercise and bile acids. And yeah, another area that has been investigated quite a bit is in weight loss surgeries. Bariatric surgeries can have very strong effects on bile acids. I would say stronger than exercise. So like Roux-en-Y gastric bypass, vertical sleeve gastrectomy, and biliopancreatic diversion all have very strong effects on bile acid synthesis and composition and transport.
Got it. So, and in general, I mean, what is the general sort of direction of the effect there?
After bariatric surgery, well, it actually depends on the surgery.
Got it. And bariatric surgery, is that the one where they're basically just making the stomach smaller?
It depends on the surgery. So,
okay.
So in vertical sleeve gastrectomy, they're making the stomach smaller. They're making the stomach into like a sleeve that is then smaller and narrower. And in Roux-en-Y gastric bypass, they are connecting a lower part of the intestine up to connect directly to the stomach. And in both of those surgeries, there is an increase in the plasma circulating concentrations, systemic circulating concentrations of bile acids, but relatively little change in the synthesis of bile acids. And after biliopancreatic diversion, it's a more extreme surgery, and people are left with only a small portion of their terminal ileum where the food and the bile are connected. So it is a malabsorptive surgery, and it also results in a very, very strong increase in the synthesis rate of bile acids because of the impaired ability to sense how much bile acid there is in the intestine. If the intestine doesn't cannot sense how much bile acid there is, then it secretes insufficient amounts of hormones like FGF19 that regulate bile acid synthesis in the liver. And so the synthesis rate goes way up.
Yeah. So given what you study in general, and given what you mentioned earlier about one of the functions of bile being a way to regulate excess cholesterol, can you give me like a bird's eye view and your perspective on the ways in which the body is sort of monitoring and deciding how much cholesterol is in the body and how much is too much and how much is too little? So for example, we talked about bile and how this is a way to get rid of excess cholesterol by basically just pushing it to the GI tract. And then you can decide if you want to reabsorb some more or not. Cholesterol, it's obviously in lipoproteins and gets transported around the body through the bloodstream for other reasons. It's the precursor to certain hormones. So like the demand for hormone synthesis probably plays into this. But like just sort of bird's eye view, as someone who understands all the pieces of biology that you do, what are sort of the main things that the body is looking at when it's deciding how much cholesterol to keep versus synthesize versus excrete? And obviously, if we could tie this to like, how much is too much and questions that might be relevant to people thinking about health and disease, that's great. But like, what are all the ways that the body is using to determine how much is too much when it comes to cholesterol?
Yeah, so cholesterol, in terms of the circulating cholesterol and the hepatocyte cholesterol, is regulated through at two major sites. On the plasma membrane on the blood-facing sinusoidal membrane of a hepatocyte, there is a very important receptor called the LDL receptor.
Mhm.
And that is the most important determinant of how much LDL cholesterol is present in the bloodstream. And on the opposing side of the hepatocyte are cholesterol transporters called ABCG5 and 8 that are the primary determinant of how much cholesterol is effluxed out of the hepatocyte and into the bile.
Mhm.
And those two components are regulated by a variety of pathways. For the LDL receptor, one of the most important determinants of its expression is cholesterol, intracellular cholesterol itself.
Yeah.
Cholesterol is also very strongly homeostatically regulated inside of cells through a very well worked out sensing pathway through the SRABP2 transcription factor response that senses when the cell, especially the ER, the endoplasmic reticulum, when the cell is deficient in cholesterol, when the ER membrane cholesterol dips below 5% of the membrane, then the SRABP2 pathway turns on and it induces the expression of the LDL receptor, and it also induces the expression of cholesterol synthesis enzymes in order to restore the amount of cholesterol inside that cell through whatever means necessary by increasing its uptake and by increasing its synthesis. And in that way, cholesterol inside of a cell is very tightly regulated.
Got it. So cells care very much about how much cholesterol is around, in particular, how much is in the membrane. Why is there such tight regulation here? Why is there such a finely tuned machine in cells to keep track of this? In other words, what is the cholesterol doing in the membrane that's so critical?
Cholesterol in the membrane is important for the fluidity versus the rigidity of the membrane. And we need cholesterol in all the membranes of our cells and all the membranes of all the cells in our body. And interestingly, the different membranes, around different organelles or the plasma membrane have different contents of cholesterol. So like I just mentioned, the ER membrane is tightly controlled to be 5% or less cholesterol, but like the plasma membrane is reported to be up to 50% of the membrane. We think of it as a phospholipid bilayer, but 50% of the plasma lipid, of the plasma membrane lipid content is cholesterol.
And so give us a sense for obviously a cell, cells are they're fluid. I think probably have a good intuitive sense for that. But what is if a cell goes from 5 to 10 to 20 to 40% cholesterol and the membrane fluidity is changing, what are the major things about what the cell can do or is doing does that actually impact?
Well, one of the things that impacts is the formation of signaling nodes, because there are structures in the membrane where cholesterol would be particularly enriched. And over the years, these have been entitled lipid rafts, and they are areas of the membrane where a lot of different receptors and proteins that respond to those receptors are co-localized to maximize the efficiency of that signaling pathway.
Got it. Yeah. So maybe it's helpful, when we look at a textbook or a diagram or a picture, it's a static image. And so that maybe gives us a misleading sense of there's a receptor in this part of the membrane, and then over here, there's another receptor, but actually things are moving around literally like rafts on a river or something. And for under different circumstances, you might want things to coalesce or come together. You might want to have different proteins and stuff all sort of attached to each other and not float away too easily or something like that. And how fluid the membrane is kind of regulates, I guess, the biophysics of how all of that happens. And so if you want to, I guess if a cell wants to modulate, where these rafts are, how easily they can form, how easily things come apart and proteins can kind of come together or float away from each other, cholesterol, the concentration of cholesterol in the membrane would be one way to do that, or the way, I guess.
Yeah.
Yeah.
Mm-hmm. Yep.
And so what, so I guess a lot of the major drugs that people may have experience with or may have heard of, they're regulating aspects of some of the mechanisms that you mentioned here that are regulating how much cholesterol is coming into the cell and how much gets synthesized. How, when we think about just like the cholesterol levels that get measured in our body, when we go to the doctor and people get like LDL measured, how much of the cholesterol in our body is coming ultimately from the diet versus how much is coming from our own cells synthesizing it? Is it mostly one or the other?
It's most of the cholesterol in our body, including in our bloodstream, is synthesized from our cells. Mm-hmm. And a more minor proportion is from what we consumed in the diet.
Yeah, yeah. Okay, so most of it comes from our own cells synthesizing it. And you said it's homeostatically regulated. So there's like little molecular thermostat mechanisms inside cells that can sense if it goes up, if it goes down, and things can turn up or turn on, and genes can get expressed and all that stuff to regulate this based on if things get too high or too low. So there's lots of sensors and stuff in the cell and feedback mechanisms that keep things in a fairly tight range within our cells.
Yeah, exactly.
Yeah. And so what if our cells are that good at regulating cholesterol, synthesizing it, turning down synthesis, all of these mechanisms are in place? Why is it so common? Like, give us a sense for how you think about why is it so common today that people need these cholesterol medications? Because, for the variety of reasons that they do.
Yeah.
Like where do things break and why, I guess is what I'm asking.
Yeah. I mean, the primary determinant of how much cholesterol is in your blood or how much LDL cholesterol is in your blood is the LDL receptor, predominantly in your liver. And so, there are a lot of people trying to research regulation of the LDL receptor, and it's regulated by a number of different pathways, and some of those can go awry due to diet or conditions like, a variety of different conditions, let's say. Mm-hmm.
Got it. So the main determinant is the LDL receptor.
Mm-hmm.
Got it. And so, okay. When we think about metabolic diseases like diabetes and insulin resistance, we were talking about that before. Normally, like when people talk about this, insulin's involved in fat storage, insulin's involved in taking glucose from the bloodstream and taking it into cells for storage purposes. Does insulin also affect in any like direct way fat and cholesterol, or is it mostly about taking glucose from the bloodstream and transforming it into a storage form?
Insulin regulates fat and cholesterol in a lot of different ways. So insulin is a primary driver of fatty acid synthesis and triglyceride synthesis in hepatocytes. Mm-hmm. So insulin promotes the production of lipid. If you think about that, that makes sense, because if insulin is responsible for clearing glucose out of the blood, it also then supports the conversion of that excess carbohydrate into a good storage form for excess carbons, which would be lipid.
Right. So in other words, if you're going to take glucose out of the blood, you either have to use it to burn it for ATP production, but if you've got more than enough, then you have to transform it somehow to store it, because we don't store glucose per se.
That's right.
Got it.
And people with insulin resistance and diabetes have a lot of changes in their lipid homeostasis, including a very classical dyslipidemia, which is characterized by increased levels of triglycerides in the bloodstream, triglyceride-rich lipoproteins, and decreased levels of HDL cholesterol in the bloodstream.
Got it. What are some of the things that you guys are working on in your lab today? What are some of the questions that you're trying to solve?
One of the things we're working on, just going back to bile acids for a moment, is a question of how insulin signaling controls the transport of bile acids across hepatocytes from the bloodstream, across the hepatocytes and into the bile. We have observed in human subjects that insulin infusion into people reduces the concentration of bile acids in the bloodstream, and this is defective in people with insulin resistance. And we can recapitulate in mice that mice with insulin resistance also have impaired ability to get a bile acid, in this case, a radio-labeled bile acid, a tracer of a bile acid across from the blood across the hepatocytes and into the bile. And this can be restored, this transport of bile acids can be restored if those mice lose weight. We also know that that process requires insulin signaling, and that insulin signaling is occurring inside the hepatocytes through the hepatocyte insulin receptor.
Interesting. And beyond bile acids, what type of things do you work on in the lab?
We love to think about lipoproteins. We do have some ongoing research projects about mechanisms by which insulin signaling controls lipoprotein metabolism. And we have a sort of newer branch of our research investigating the consequences of the divalent cation manganese on insulin signaling and metabolism.
Manganese. So that's that's a nutrient, and it's one of the ones you don't hear about quite as often as some other ones. But what are some of the basics there you can tell us about manganese and what it does for those that don't know?
Yeah. Manganese is an essential micronutrient. We need it in all of our cells. It's an important enzyme cofactor, including a variety of different known enzymes. But there are likely other proteins and enzymes that are affected by the concentrations of manganese that are yet to be understood. Manganese is also very strongly regulated because it also can be cytotoxic in high concentrations. And one of the reasons that we became interested in it is because it is also similar to bile acids, primarily excreted via the hepatobiliary route through a transporter that is on the canalicular membrane of the hepatocytes. Yeah, so those are sort of the basics, I guess.
Yeah. Well, where do we, are there foods that tend to have manganese in them? Where do we normally get that in the diet?
Manganese is present in a lot of different foods.
Yeah.
It's enriched in like leafy greens and lots of different foods. Yep. But it is quite difficult to increase a person's manganese content simply by diet alone because the excretion is so efficient.
Oh, I see. Okay. Got it. So what other things are you working
on? We have some projects about bile acids in liver disease. So a variety of different inflammatory liver diseases have changes in bile acids, and we have been investigating the roles for different kinds of bile acids in the inflammation and liver disease progression.
Yeah. So in terms of something like fatty liver disease, that has in many ways become more common over time. In particular, that's something we see now in children. Like whenever I've heard people talk about fatty liver disease before, one thing I've often heard is like, we used to call it alcoholic fatty liver disease because we'd only see it in like lifelong alcoholics. But then we basically had to rename it because we started seeing it more and more often as time went on. And now we see it actually fairly often. I forget the numbers, even in fairly young children. What's your general perspective on like what's driving that beyond just like our diet sucks? Is there something in particular about the modern diet that has pushed that to earlier and earlier ages? Or like, what's your bird's eye view there?
We are surrounded by an environment of overconsumption, and think it is entirely possible that the sort of processed foods of today are contributing to the development of what we, as you just alluded to, we now call it the metabolic dysfunction associated steatotic liver disease. And the condition of insulin resistance is also a major contributor to the development of MASLD. And so as people become more insulin resistant at younger and younger ages because of their diet, because of obesity, and things like that, then they are more and more at risk of MASLD.
Mm-hmm. Are there any final thoughts you want to leave people with, or anything you want to reiterate from our discussion today, particularly as it relates to liver function and bile acids and health and disease?
Final thoughts. I think bile acids are very fascinating. They have a lot of different roles, and they are particularly known for their role in emulsifying, so their physicochemical properties, their physical ability to emulsify things. But even at low concentrations, those bile acids can act as signaling molecules binding to a wide variety of different receptors and activating signaling pathways inside of a cell. And so I think they continue to be very interesting. We as a field are discovering more and more different types of bile acids and different types of bile acid modifications that are present in the human body. And each one of those modifications can then affect how well those bile acids can carry out their functions. And so I think there is a lot of research in the future to understand the consequences of changing bile acid composition, concentration, and transport.
All right. Well, Professor Rebecca Haeusler, thank you very much for your time.
Thank you so much, Nick.
A great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio, formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.